I'm very excited to introduce Professor Willem Hanekom to give a lecture on TB vaccines.
Okay. Yeah, thank you. Thank you for that introduction. I thought I'd start off by telling you a little bit about IDORI, in case you want to work with IDORI. We are a strategy-driven organization. So our vision, so what we want to achieve is optimal health and well-being of under-resourced populations. So we only work with really very under-resourced people. And to do that, if I can jump straight into a scientific strategy, we address complex health problems, and we specifically embrace the complexity of these health problems, because in under-resourced populations, in individual households, there are often multiple of these problems in the same household. And we address these with multidisciplinary approaches. And we have three main health areas. They obviously intersect, as I've just said. By far the most work is in infection, mostly HIV and tuberculosis, and also ancillary conditions. So, for example, NCDs associated with HIV. Adolescent mental health, we started a new program specifically when we try to understand how the communities around us view well-being. And when we went there and said well-being is having food on your table, money in your pocket, a roof over your head, what are the issues? And everyone talked about adolescent issues. So that's why we started this program. And climate and health should be pretty obvious. So the multidisciplinarity ranges then from basic sciences to clinical trials, data science, implementation science, epidemiology, indigenous knowledge medicine, and social science. So in the end, we work in two campuses. That's where we do our work. The one is the fancy building in Durban, and the other is a very rural area where by far the majority of our activities happen. And there is a health and demographic surveillance system, which I know there are two of them here in the Johannesburg area. Also, when one characterizes a population in extraordinary detail and then uses that as the foundation to do whatever research one does. We're an independent company, a non-for-profit. We have university partners, but we're not affiliated, and as such, therefore, we have a huge business arm to be able to do everything that we do. More than 800 people and 33 faculty members. We really have a huge emphasis on our personnel. This was at a year-end party when people always dress up like you cannot believe it. So huge effort to look at culture, and that's one of, I think, the proudest achievements that I have at IDORI is people are happy to work there now. But ultimately, it's all about the impact, and really the two outcomes that we measure are really critical knowledge, because not every molecule discovered in the lab is going to translate into policy and practice. That's why we have that separate also, but we have many examples of how that happens. So that's a little introduction of IDORI, and if you're ever interested about it, go to ri.org and come and visit anytime. So today about TB vaccines. I'm going to present a hodgepodge of things. I'm not really going to present primary research. It's really going to be an overview of what the field looks like today. So I'll start a little bit talking about TB and TB control, and then the current and novel TB vaccines that are in the pipeline, and then innovation, how to get to where we want to ultimately get. So TB is normally a lung disease, okay? And if you are not a medic, what you see there is X-ray. In the middle, you can see the shadow of the heart. And on my side, there's muck in the middle. And that shouldn't be there. It should be cleared like the other side. And that is in this patient, tuberculosis. So tuberculosis has classical symptoms that come on gradually, such as coughing, chest pain, tiredness, loss of weight, decreased appetite, and often night sweats. So normally, up to now, we've identified patients with TB by looking for these symptoms, but what we're now discovering is something completely different. And that is that more than 50% of persons with TB disease may be completely asymptomatic. So this is, it's extraordinary that we're only discovering this. We've discovered this in the last 5 to 10 years. And this, of course, has huge implications for how one identifies people with TB, how one treats them, what you want vaccines to do, etc. So I'm happy to talk about that if there are any questions. But as such, if you just look at TB disease without asymptomatic disease, it causes 10.8 million deaths in the world every year. And that is the #1 identifiable infectious disease killer in the world, therefore, more than HIV, for example. And in South Africa, it's 10.8 cases, people with disease, not deaths, and then 270,000 in South Africa become sick with TB. And 1.25 million people die from TB globally, and about 56,000 in South Africa. And so that may mean absolutely nothing to you, but in the stadium, where Kaizer Chiefs beat Orlando Pirates on Sunday, or Saturday it was, that stadium takes exactly 56,000 people. So if you go to that stadium and go to a soccer match, then you'll know exactly how many people die from TB every year. Huge, huge amount. So in terms of TB control, if you look on the left at incidence, on the right at death rate, so there's been a gradual decrease. That's been very good news until we hit COVID. And of course, with COVID's restrictions, TB interventions became less, and there's been an increase in the incidence globally since then. And similarly in the death rate. And South Africa has sort of followed the same trends. So it's a huge problem, completely not under control. And to control this, we have drugs. We have drugs that work very well. We've got very good diagnostics, but somehow we're not doing enough to control this epidemic. So that's where vaccines come in. So we have a vaccine. This is a child from Satvi, where I worked before, as Glenda said. We've got a BCG vaccine on the right arm, which virtually every baby in the world gets soon after birth. And this is a very good vaccine. Okay. BCG is about 80% protective against severe disease such as meningitis, about 60% protective against lung disease in young kids, and perhaps about 20% protective against infection itself. So it's a very, very good vaccine. But regardless of the BCG, and BCG has contributed around about 12% of the 152 million people saved by vaccines over the last 50 years. So BCG is a fantastic vaccine. How did that happen? But anyway, the issue is that it does not prevent disease in adults and adolescents. Now, in TB, you have a bimodal presentation of TB. It's common in young, and then it becomes more common in late adolescence, and men obviously have more TB than women. So when adults have TB, or adolescents, they have a different kind of disease. They have big holes in the lungs, very inflammatory, lots of bacteria, multibacillary. They spread the disease. So unless we get a vaccine to target adolescents and adults, we will not control the TB epidemic. And there's this beautiful modeling done that shows that you can think of anything, identifying new TB cases, whatever, but ultimately you need a TB vaccine that prevents disease in these age groups to control the TB epidemic. So the WHO has preferred product characteristics published for what we want a TB vaccine to do, and so they've included vaccines against targeted at infants and children, and vaccines for adolescents and adults. And I'm only going to concentrate on the latter because I think we've got a fantastic vaccine. We don't need a replacement for babies. And the bar is very high to show superiority over BCG. So the important thing is that the modeling shows that even 30% efficacy will, against adolescent and adult disease, would have a massive impact on the epidemic. So the PPC asks for 50% efficacy, but it's very clear that we don't need the 80-90% efficacy that we needed in COVID, for example, to do something about the epidemic. So in terms of new vaccines, then, there is a pipeline. Okay, so phase 1 studies is really about safety and some immunogenicity, and then you go into the target population, phase 2A, and then the first efficacy, preliminary efficacy with wide confidence intervals, usually in phase 2B, and phase 3 is more definitive efficacy. So you can see there's quite a lot of candidates in the pipeline, but think of COVID, where there are literally hundreds of candidates, and I don't think TB is any less of an issue compared to COVID. And also, the other thing that's very, very unusual here is how the pipeline is skewed to the later phases. What you really want is vaccines in early phases, lots of vaccines in early phases, then because vaccines do fail in their clinical testing ultimately. So I'm going to, rather than going through these one by one, I thought I'll give a sort of summary overview of what they are. So there are different kinds of vaccines. Some of them are whole cell vaccines. So BCG, for example, is a Mycobacterium bovis, okay, which has been attenuated by serial culture in a lab more than 100 years ago. And so there are other such whole cell vaccines also, which are all in blue here. And then you've got protein vaccines with adjuvants, and there are quite a few of them, some viral vectored vaccines, and the only viral vectored vaccine would, in the entire, or both these viral vector vaccines are the only 2 vaccines that are currently being given by mucosal route. All the others are parenterally given. And then 2 mRNA vaccines from the same company. So if we had to break it down even further, we've got an undoubted most advanced and promising vaccine, which is called M72, which I'm going to talk about a little more. And then we've got a fast follower, as I would call it, called MTB-VAC, which I'll also mention a little bit. And then I'll also go into some of the newer generation vaccines. And I'll say why I think they are sort of newer generation vaccines. But before I go there, this is what I meant, is that product development is a risky undertaking. And it does fail at times. So, for example, recently there were 3 candidates that showed negative results were shown from clinical trials. So, in a prevention of infection, so what we want in vaccines is to prevent disease, okay? But you may also prevent infections. For those that do not know, if I have TB and I go around cough, 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 cough all over you, or even breathe, we know. Okay, all over you, quite a few of you will become infected, but by far the majority of you will control the bacterium. Only 10%, usually 5% in the first 2 years of those that become infected will develop disease. So you can have a vaccine that can prevent infection also, but the problem is you don't know if the prevention of infection was in the 90% that were never going to get disease. So it's a difficult endpoint to actually measure, but regardless, BCG was in South Africa. Actually, this study was done where adolescents who were quantiferon negative, so that they hadn't seen TB before, were given BCG, and then they were followed up with serial interferon gamma release assays, IGRAs, quantiferon tests. And they basically, although the first study showed protection, the more definitive, larger sample size study showed no protection. Similarly, one can use a vaccine in prevention of recurrence. Now, in high prevalence areas, such as this, when you have tuberculosis, there's about a 4 to 8% chance of getting disease again within 2 years of having had clinical and microbiological cure. So that could either be relapse from the original strain where you thought the person was cured, or it could be reinfection. And maybe, and I think this is what's happening, is that people are compromised in some way or another, and that's why they get reinfected and get disease again. But 2 studies recently with one subunit vaccine and one whole cell vaccine, a recombinant BCG, has shown no effect in preventing recurrent disease. In fact, age 56, IC31 may have made relapse worse by giving it at that time. So it's a risky undertaking. But let's start then with M72. So M72 has only 2 antigens of possible 4,000 protein antigens that tuberculosis could have. So why would a vaccine with only 2 antigens work? Perhaps a lot of it's in the adjuvant, which is called AS01E. And it's got MPL, a TLR4 adjuvant. It's got a saponin called QS21, which comes from the soap bark tree, and you can see the flower there. And it's formulated in a liposome. And so this vaccine is currently in a phase 3 trial that I'm going to talk about a little bit right now. So the good news was that in a phase 2b trial, so the first trial to show efficacy, or where we look for efficacy, we showed 50% efficacy in protection against TB disease in persons who were quantiferin positive and who got the vaccine. So it's that you can see there. The important thing here is that there was a sensitivity analysis done also, and this sensitivity analysis had more strict endpoints than the ones in the original study. In fact, I think much more relevant endpoints. Okay, and there the protection was 68% with a lower limit of the 95% confidence interval of 25%. Now that is licensable based just on the phase 2b results. So why wasn't it licensed? Because the manufacturer or the sponsor didn't plan for success. There was no product available. There was no that's the one part. The other part is that one also needs to get results in very specific populations, such as persons living with HIV and quantiferin negative populations, which wasn't available, and therefore this could not go for licensure, and therefore one had to go on to a phase 3 trial. Now this trial, which costs $550 million, so I'm asking you, how many of these trials are we going to do? I'm going to be affording, especially in the current climate that emerged this year. The indication is really prevention of disease. And in quantiferin positives, in quantiferin negatives, you cannot get to a sample size of persons living with HIV that can practically be done. Therefore, number 2, one just does safety and immunogenicity, and hopefully the regulator will accept that as good enough to actually carry forward. But this study has 18,000 persons from various South African and Southeast Asian areas who get the vaccine or placebo, and we hope to show 55% efficacy. And it requires 110 cases. So far, everyone has been enrolled and randomized, and we have about 15 cases of tuberculosis so far, about 2 years and a half into the study, and we will probably have results by 2028, and I'm absolutely confident this is going to be a positive result. But 2028 is round the corner. And we do not want to be in the situation that the malaria vaccine was in. It's called RTSS. I cannot see there. Was it 2015 or 20, when was that? 2015. Yeah, 2015 the efficacy results came out. Guess when it reached people? Only 2 years ago in West Africa. It started rolling it out. We do not want to be in this situation, so there's urgent work that needs to go on right now to plan for success. So a lot of that work is happening in South Africa. So what I'm saying here is you cannot just do a proof of efficacy in a phase 3. There are many other steps that one has to get through to ultimately get a vaccine to where it needs to be. You know, you need to procure it. It needs to be made. It needs to be procured. People cannot be hesitant, etc., about the vaccine. So they're beautiful documents, and you're not supposed to read this, but it just shows you all the steps that one needs to go through. And a lot of these things are happening in South Africa. And I was hoping, and I couldn't get it in time, but I saw a presentation about confidence and hesitancy, specifically relating to TB vaccines in South Africa. And the hesitancy rates were in the 40% range, okay? So, and very beautifully done studies. So we have a lot of work to do here. And so in South Africa, there is a consortium that's already funded and is doing lots of the preliminary work to see how this could be integrated into the healthcare system, also addressing issues such as confidence and hesitancy. But perhaps one of the most important structures that are working on getting the vaccine to where it should be is a WHO committee called the Vaccine Accelerator, which has this roundtable thing full of very important people that just meet and say the right political things, and then other people need to do the work. And so the work is the kind vaccine research is not really in the remit, but really to make sure that that the product development of the individual products could be done as efficiently as possible to be able to get the approvals it ultimately needs. Similarly, the financing and access is a huge issue. So there's an established working group that's looking first at the demand of this vaccine for ultimate use within the world, M72. And then there's also a group that looks at country readiness, advocacy, and community partnership. So, for example, for that, the WHO is, with South African stakeholders, is having a stakeholder meeting in July to look at this. But the bottom line that we have right now is that we may have results in 2028 that show fantastic efficacy, and we may have no product. And the reason is because a commercial partner for this vaccine has not been identified by the sponsor. It hasn't been signed, and everyone here who works in product development knows that tech transfer takes time, takes years and years. So we may already be too late to ultimately get the vaccine in time. And at the core is this. Shingrix, the shingles vaccine, contains the same adjuvant as M72, and it earns GSK $4 billion a year. Now, we know that we need an investment, an at-risk investment of $200 million to make a facility to make enough AS01 for TB vaccines for the world. But GSK, even though they earn $4 billion a year, and I shouldn't be negative, thank God, there's one of the companies that is still in global health, okay, but is not prepared to make this investment. In fact, no one's prepared to make this investment. So we're in quite a dilemma in terms of TB vaccines, and therefore, right from here, I think we need contingency approaches as soon as possible and as urgently as possible. So one of them may be the other fast follower. So MTB-VAC is, so this is how we got BCG. I mentioned earlier, serial culture from a cow that was sick. And that's what we use in children now. Okay, and so MTB-VAC, though, is a virulent strain of M. tuberculosis, one of the lineages that was identified, and only 2 mutations, okay, allowed this vaccine to become as avirulent as BCG, in the lab at least, and now it is in clinical trials. So this is already in a phase 3 trial, phase 2B trial, so the first efficacy trial in South Africa. In babies, it's in much later trials already. So this is at least one contingency, and they have a much better manufacturing strategy. For example, they already have 3 partners in 3 different continents to make this vaccine. So this is a good thing that they've done excellent in a good way. And then we've got some next generation vaccines, and clearly mRNA vaccines are the prototypes of these because of this vehicle to bring the vaccine to where it needs to go. So the BNT, the BioNTech vaccines, there are 2 of them. The B1 vaccine has the same backbone as the COVID Pfizer vaccine, Pfizer-BioNTech vaccine, okay? Whereas the A1 vaccine has an unmodified uridine, so it's more, it's got greater immunogenic effects, especially on the innate immune system. So whether that's going to be safe, we'll still have to see. But also the other innovation here is that suddenly, rather than the other vaccines, the earlier generation vaccines such as M72, there's a range of antigens that have been chosen very carefully based on various roles that they may play in either keeping latency. We don't talk about latent TB disease anymore. We talk latent TB infection. We talk about TB infection and TB disease. But to maintain that latent infection, these antigens may be important. For reactivation, this antigen may be important, and then there's some immunodominant antigens associated with clinical disease. So this vaccine is now in a phase 2A trial in Southern Africa. The sponsors are doing a fantastic job at rushing this, and I think this is, it may even have results at the same time as some early efficacy results at the same time as MTB-VAC, if it's going at this rate. Another example of a next generation vaccine that also has learned from what we know about antigens is a subunit vaccine called H107E. Here it has repeats of ESAT6 immunodominant antigen. There's various other antigens also very rationally selected, and then an adjuvant that interacts with Menkel and TLR9 in the innate immune response. So rationally designed, induces very careful studies to show that it induces Th1 and Th17 cytokines, the kind of cytokines that we think that are important for ultimate protection. And this study is now with Orem in a phase 1 first in humans trial in South Africa. So there is some next generation vaccines that are on its way. But what more can be done? And so what I thought I'd talk about is first populating the pipeline. So it's very clear that we need more candidates, ultimately. So we start off with this. So the current TB vaccine R&D funding amounts to about 18% what is estimated to be needed to get to a vaccine ultimately. And these are the sources of the funding: NIH, Gates Foundation, Open Philanthropy, and Wellcome, and others. But this is the situation that we're dealing with. So for example, we have in TB vaccines about 1/7 of what HIV vaccines and BNABs, etc., have in terms of development. So there is an advocacy aspect that's very important. But also, if you then look at the candidates that are in preclinical development, this is just a selection that is on the Stop TB Partnerships website. You've got a slew of whole mycobacterial candidates. All right, you can see a lot of them, and then one viral vector. There must be others, though. Also, there's another subunit vaccine, and then various nucleic. I know of at least 3 groups that have nucleic acid vaccines that they're moving towards the human at the moment. So one of the problems that we have here is that we don't have animal models that are reliably predict what is going to happen. So our product development stage gating is extraordinarily difficult. I think there are now efforts to compare all these whole cell vaccines, and they've already been done by a group in Australia. But again, the models may not reflect what actually happens in humans. In fact, for M72, we were talking about this over lunch. For M72, if you just looked at the animal results, the NHP results, I'm not sure you would have gated into humans. And now it's the most promising candidate. So we don't quite know how to do this. There's a lot of work. But anyway, if you look at the antigens in the vaccines, so these are the ones that I showed. I didn't show ID93, but I showed the other two in the boxes, and they're modern in terms of the antigen selection. But all the other candidates that are subunit viral vector mRNA or viral vector vaccines have immunodominant antigens, antigens that have just by screening all of us here, if we've seen TB, taking our blood and seeing which antigens we react to with T-cell assays. And that's basically it. Now, is that the right way to go? Perhaps not. Because if you look at immunodominant antigens such as these, all over the world, if you take strains of MTB from all over the world and you sequence them, and you look at what happens to the immunodominant antigens, you can see very, very little variation across all these. So why isn't there so much variation? Whereas if you look at other antigen groups within MTB, there's much more variation. So this ultimately, if you go through the arguments, you can actually say perhaps the MTB is using immune responses against these antigens to its own advantage, ultimately from an evolutionary perspective. So perhaps we should be looking at other antigens, but can't we just use whole cell vaccines, and therefore that slew, as I called it, may be completely appropriate? And perhaps not, because BCG, for example, we know that if you give BCG to babies, small babies, it works. But as soon as that baby is at significant mycobacterial exposure, be that TB or other mycobacterial exposure, the vaccine's efficacy drops dramatically. So I don't know in our settings where we need vaccines for adults, whether MTB-Vac or any of the other whole cell vaccines will work. They induce immune responses on top of previous exposure, but whether those immune responses are the right ones for protection, we don't know. So I think we've got a lot of work to do to decide which antigens should be used, and we had a special meeting at Sanger Institute to talk about this for 2 days and come up with specific plans which are going to be very expensive, and already the funders have said they won't fund that. So, but anyway, some of the things that one can do is go to cohorts, okay, and make sure there are cohorts with sample collections. So some of these cohorts are correlates of risk cohorts. So if I follow a huge group of people and see some of them are going to develop TB, by far the majority over time in a high prevalence area, and the majority not, and then look at samples collected prospectively to see what are the correlates of risk. Those may be tips for looking for correlates of protection. And in fact, that's exactly what Tom Scriber and Munya Musvosi did in Cape Town. My only claim to fame is that I set up this cohort. So that's probably the only thing, but they took it to the next level. It was 6,363 adolescents that were followed longitudinally, and they had bloods collected over time. We could describe immune correlates of risk in these, or signatures of risk, mRNA signatures of risk. But here they looked at the actual T cells in those that developed active disease, which was 48 of them, and they chose 118 controls. All right, and what they then did is they compared TCRs, and clearly we think that the controllers had good TCRs, and the ones that didn't control had bad TCRs. All right, and now you cannot compare just individual TCRs. There are too many, and there's too much variation. So they were able to look at TCR similarity groups in a technique that I'm not going to go into, but which was described by Mark Davis in San Francisco first. And they could clearly identify some TCRs that were associated with control and some TCRs that are associated with progression. And then with very fancy reporter systems, you can then actually ultimately identify which are the antigens that bind to these TCRs. So some antigens have been discovered. Here are two of them. The one is PE13, and another one is an immunodominant antigen called CFP10. And working with Patrick, who's sitting right there, and others, they've already constructed mRNA vaccines. So this is, for me, the first rational vaccine or antigen incorporation in vaccine ever in the TB world from clinical data that has happened. And so it's very exciting, and I'm hoping that this vaccine, or hopefully not both, but at least the one that you choose ultimately will be chosen soon and go into clinical trials pretty soon. So again, once you have cohorts that you can do these kind of studies, you can get much more rational antigen identification. Again, correlates of risk. We hope that correlates of risk will translate to correlates of protection. All right, another example of cohort studies is, so first let's look at what the many components of the immune system that are activated by tuberculosis. But as I've said earlier, there's probably two groups of T cells that make Th1 and Th17 cytokines, and then cytotoxicity that has been associated with protection over and over in animal models. So perhaps those are important, but now that we have samples from this study where there's protection, we can actually do correlates of protection studies, and a huge such effort is going on, led by Lisa Nemes in Cape Town at SATVI, where many different approaches are being tested. They first validated the assays, made sure there's an adequate range to actually look at differences and look at the performance of the assays. And they're now already busy with the analysis to try and identify correlates of protection. Obviously, as soon as you have a correlate of protection, your clinical trial, and that applies across vaccines, your clinical trials could be much more efficient, or your testing of new products. Another cohort kind of approach that is happening at IDORI, where I'm working. So this is what the area looks like in the north where we work. And so we do autopsies in these areas. We've got a professor of pathology that's one of our personnel members, and he does autopsies. And what one then does is identifies lungs, or get lungs from people who die from other causes, not tuberculosis. And because some people have TB infection that's well controlled, like 90% of the population, perhaps those hold the clues to what you want in terms of protective immunity, right? So the good thing is that they've shown now over and over and over that they can actually identify with fancy tomography putative TB lesions, and then go to them and show the proteins in these lesions. So these are TB lesions. So this is now in a major new effort, grant with Audrey and Tom Scriber and others are going to try and look at host responses that may be associated with protection. Then the NHP, for all the criticism I gave earlier, can give us incredible results. So if you give BCG IV, and I wouldn't recommend that you ever do that to any human, because it's a live bacterium, and people are probably going to get very sick and may even die. Some people may even die from it. But in the non-human primate, BCG was given IV, or the primates were left unvaccinated. they were given intradermal dosage, low or high dose of BCG, or aerosol BCG, or aerosol with ID. Okay, and you can see in terms of these total colony-forming units, the amount of bacteria in the lung, how effective IV BCG was. And in fact, if you look at PET CT scans, you can clearly see the row that got IV BCG. Disease here is in red, that you can see on the PET CT scans. So what they then, this is a fantastic model. We're not saying we should use that in the human, but now they could down titrate the IV BCG at lower and lower dosages to get breakthrough disease, and then it becomes a correlates of protection study. So I'm just going to, without showing primary data, I'm going to show you what they saw. They were able to incorporate in these multivariate models T-cell responses, cytokine responses, antibody responses in both the lung, BAL, bronchoalveolar lavage, and blood, and then do this analysis to see which things protect. And basically, virtually everything you looked at in the lungs looked like correlates of protection, okay, but very little in peripheral blood, all right? But the ones that, again, like that that was most strongly associated with protection was CD4 responses and NK cell responses, especially cytotoxic responses. So again, my proposal, and from that meeting that I mentioned in Sanger, is that we need to take all this data together and put it in models, maybe AI-driven models, to be able to actually get it to TB antigens in a rapid manner that we should ultimately be using. Regardless of what I'm saying in terms of getting to antigens, is that ultimately it will need an investment for GMP manufacturing and testing these antigens in humans, which is the cost. All right. Okay, I'm going to end off with just one little aspect of innovation. So product development can be much more efficient than we do it currently. We can use AI tools. We can on many levels of the clinical aspects, even in the labs, and how you design your experiment from day to day. You know, how you do your statistical analysis, etc. And so this is the new direction in which we're going to go. But in clinical trials, clearly more efficient trial designs, approaches than we currently have. Ultimately, there's very few shortcuts, but at least we can try that. But there's also other tools that are coming along, such as human challenge studies. Now, human challenge studies are used very extensively in malaria, for example. So when you give a vaccine to someone and then you get mosquitoes to bite that person, give that person malaria, and you see whether the vaccine has worked or not. And of course, you use a malaria strain that is imminently treatable. So would you do this with TB? If I inject you with TB after I've given you a vaccine? I'm not so sure. It's a different kind of, it's a chronic disease. It causes, so even after you're cured from TB, you have a 2 to 3 times higher risk of death. It's a really devastating bug. So can't you just develop strains to do that? And yes, and so people at Harvard, Sarah Fortune and others have developed such strains. So these strains have triple kill switches. So they say they're under antibiotic control. So here it's doxycycline and trimethoprim, what is it? Bactrim, whatever. And so while the mouse is getting the antibiotics, the strains grow. But as soon as the antibiotics are withdrawn, the strains do not die immediately. And so now we can use this, hopefully, as a model to do human challenge huge regulatory and other issues to get past, but hopefully we will soon have human challenge that we could use also to try and down select or up select some of our vaccine candidates for the future. I have to say, though, this is different from an acute disease such as malaria or gut disease such as infections. That's the other area in which challenges are happening routinely. This is a chronic disease, tuberculosis. So all we can really look at is the early stages after infection, whether these can be controlled by the vaccine if we had to introduce such a model. Okay, so my conclusions are we need vaccines that prevent clinical TB in adolescents and adults, and perhaps also asymptomatic or subclinical TB, but we don't know yet. Okay, M72 is in the phase 3 trial and showing incredible promise, but there are issues that I mentioned. Manufacturing, are we going to have the product available when it needs to be there? MTBDAC is the fast follower. As I said, we still have to see whether in a phase 2B it's going to actually work. So then there's a range of next generation vaccines that are emerging. They're based on science, advanced understanding of antigens, novel platforms or optimal adjuvants. Then efforts towards innovative trial designs and acceleration, such as perhaps using challenge models. There's also efforts to work with people deprived of liberty, people in prisons or healthcare workers, and doing these studies in settings where there's much higher force of infection to get to endpoints much easier or quicker. And then finally, there's an urgent need to populate the clinical pipeline from the left. So thank you very much. Thanks.
So the BCG is a really good vaccine in babies, and you mentioned that the loss of efficacy is probably related to the infection rate. So that there's obviously there's waning immunity, and the infection assault overrides the ability for the vaccine to be protective.
So can I just interrupt there? So it may also be that as soon as you have antimicrobacterial immunity on board from, so if you did not get BCG as a baby, and you get BCG at 5 years of age, and you got infected with MTB at 3 years, you have some antimicrobacterial immunity on board, and that is enough, not allowing the BCG to take and to have its effect ultimately.
Okay.
Yeah.
So that's why you need, so it's not a durable, and there's probably different mechanisms like you suggested, which is fascinating. My second question is, you mentioned that the adjuvant in M72 might have been the thing that drove efficacy. Did anyone think about designing a vaccine just with the adjuvant?
Yeah, so from if you, and you understand product development more than anyone in this room, but after you asked us that question, that's never going to happen from a vaccine developer's perspective. They're never going to, once you've got an efficacy signal from a product, you're never going to take one aspect of that product and take it out. But a lot of people think that this may be the reason. So remember, when you are, say, 4,000 MTB proteins, you already have been infected. You may have immune responses to multiple proteins. So, but that adjuvant is enough to modulate the immune response in the direction that you want to induce protection.
Yeah, so it is fascinating. So maybe we could design more adjuvants that had that action.
Yes,
and evaluate that.
So there are many. So because of the risks of M72 and in terms of production, there's a very similar adjuvant called LMQ that is now being fast-tracked, and basically they need to be synthetic. As soon as you have non-synthetic things like MPL that you've isolated, so Bactri and whatever, then it's problematic and it's expensive to do.
Yeah. Okay.
Willem, thanks for that talk. So, I mean, just first an observation, and I think we spoke about this probably 25 years ago, is this notion of 50% of TB disease being asymptomatic. And one of the observations that we made, and in fact David Moore did it as part of his MMed, is this observation where with pneumococcal conjugate vaccine, and I think you know about the data, where we show that by giving children pneumococcal conjugate vaccine, we reduce hospitalization for culture-confirmed TB.
Yeah.
And in many of those children with culture-confirmed TB, they were their symptoms were resolving within 2 to 3 days, which is in keeping with an acute pneumonia. But it was obviously the underlying tuberculosis that probably predisposed to that, caused some sort of immune suppression and predisposed to that pneumococcal pneumonia. So this whole, it's a really important, the question is, what do we make of that asymptomatic so-called disease? I'm not too sure whether we can call it asymptomatic disease per se. It's infection, but what do we make of that? So that's the first. But the question more is, you seem to be fairly optimistic of messenger RNA as a pathway towards the next generation of TB vaccine. But at the same time, what you do indicate is that what we're looking for is a Th1 dominant type of response. And messenger RNAs have not been that great in relation to inducing Th1 responses. They do induce Th1 responses, but even if you look at the messenger RNA and the vector-based vaccines with COVID as an example, the vector-based vaccines induce much more of a Th1 dominant response. So how do you reconcile that messenger RNA? You can be more selective in terms of the type of antigens that you're selecting, obviously, and go for combination antigens, but why do you see that as being a promising sort of pathway to the next generation?
I don't know, but I think when one needs to do the studies and see what happens with these, and clearly the large companies that have huge investments into the LNPs, etc., are modulating these in the directions that may be important. So I know I'm not supposed to know this already because the data hasn't been shared publicly, but for example, the BNT vaccines apparently have been incredibly immunogenic in terms of inducing Th1 responses in the initial. I think for me the biggest issue, but again I hear there are ways around it, with RNA vaccines is the duration of protection. At the moment, the duration for most RNA vaccines are very short, and really we want longer durations. Now, going backwards to your other questions. So the first thing is, so when you're a pediatrician, when you go into wards and you diagnose people with TB, that's usually that they had a super infection that brought out the TB. That's the classical way. So I actually, and so I think viral infections and bacterial infections, such as pneumococcal infections, may actually precipitate TB to become worse. And there's pretty good evidence for that, in that those correlates of risk that I mentioned, the gene expression signatures are type 1 signatures. It's exactly the same signatures that you see from viral infections and even from pneumococcal infections. And so these become dominant in the immune response, and somehow that ultimately manifests as TB disease then. So yeah, I wanted to do, but I didn't get funding for it, but I wanted to do a study of large-scale influenza vaccination to try and prevent TB, because I think that's how there clearly has to be interaction. Now, the thing about asymptomatic TB, it's not infection, it is TB disease. So, there's now groups that have gotten together and generated definitions, and I think by far the most common definition of asymptomatic TB would be culture or positivity or a nuclear amplification test like GeneXpert positive sputum in a person who has no symptoms or otherwise was not aware that they had symptoms. So again, if you work in the field, everyone's coughing a bit, they don't think that's important, etc. Perhaps there may even be symptoms present. But so that's the one definition. And then, of course, the complexity comes in. It's that there may also be radiological abnormalities that you can detect if you actually go there, because that's not a routine test that is done in most clinical settings in South Africa, for example. So it is actually disease. And so from the results that we have right now, it seems like 30 to 40% go on to symptomatic disease ultimately. But it also seems that some may self-cure. So it's an extraordinarily difficult issue to address, and there are massive studies that are going to go on. We've just submitted a $25 million grant to do this on an incredibly large scale in South Africa and Indonesia, to look at all the variables surrounding subclinical TB. So we don't have the answers, but I think it's a very significant disease state that we need to understand better.
Bhavesh Khanna, School of Pathology in IDORI. Thanks, Willem. It's always nice to see all the thoughts together. So, almost in listening to you, whenever we hear about TB vaccines, you hear we need a new vaccine, but it's almost like, will one vaccine do everything? So will one vaccine protect adults and adolescents in all geographies? Will it protect vulnerable people? And maybe we need to be looking more at a portfolio of vaccines to ultimately get the public health impact you're looking for later on. And so I guess the first question is, what is the thinking around that, and having a multi-pronged approach that perhaps will get some vaccines to market better in vulnerable populations or specific geographies, as opposed to looking for a single vaccine that does everything. And then a second question, I've seen this come up recently, the use of microbiome and microbiome signatures as a measure of vaccine efficacy and immune responses. Are we looking at this in TB?
Oh, okay. Second question, I know nothing about that, so I cannot answer about that. First question, yes. Thank you for mentioning this. So that is part of, if you just think about the number of people in the world that needs new vaccines, it's in the billions range, okay? So there's no way that even one vaccine will suffice in any case, even, just from a, if I can call it practicality, from a access, manufacturing and access perspective. So clearly we will need multiple candidates, and that's why the ongoing development is so critical. The development becomes more complex as soon as M72 has, if it shows efficacy, now everything will probably have to be tested against M72. It doesn't mean that it shouldn't be tested, but yeah, it's a different kind of product development.
So now we have a vaccine, M72 is 30% efficacious, and obviously if you take it to scale and you vaccinate, it has an impact on outcomes. But if you're designing another trial, would you drop the placebo arm and replace it with M72?
Definitely. Yeah.
And the studies just get bigger and bigger.
Yeah.
Okay. So that's interesting.
Thank you, Prof. That was a wonderful talk. I'm David Moore from Wits Pediatrics and School of Clinical Medicine, and IDORI as well. It's fascinating encountering or being confronted again with the story of TB and TB immune responses. And the story that you painted today is really that natural TB infection seems to be its own best vaccine, with a 90% vaccine efficacy, 10% of infected people progressing towards active disease. And the comments that you made about cohort studies investigating the immune responses which keep the infection at bay, I think is very crucial to our understanding of seeing how to control the pandemic. So are there more efforts going to be made into establishing these cohort studies and characterizing immune responses in greater detail? Just as an illustration, when I was busy with my PhD work, I had an ELISA, early spot done, and I'm riddled with tuberculosis infection and haven't progressed to disease at this stage, simply because of working in a high force of infection environment, in the health care environment in South Africa.
Yeah, so thanks for that question. The first thing I wanted to comment on is, does MTB infection protect you against disease? So I think nowadays no one will do the study, of course, but there's historical studies from before the antibiotic era, where persons who were tuberculin skin test positive, so presumably infected, if they got re-exposed to TB, they had an 80% lower chance of developing TB disease. So infection definitely does protect you against future disease, but we still have the epidemic. Okay, so it's relative. So then the issue about cohorts, there are many such cohorts. There are what you may have, there's some people call them resistor cohorts, where people get exposed repeatedly to people with TB and they do not develop TB, or they don't even become infected, convert their quantiferons or other egress. So that's such studies are ongoing, and in fact one of the new antigen TB antigen identification efforts is based on such a study that happened in Central Africa. Difficult studies to do because they're longitudinal, and you have to account for exposure that happens in the interim in any case. The second thing that I'd say about cohorts, it is completely inexcusable what 2 of the phase 3 studies have done. Now, for example, the studies with VPM 1002, where they have no samples collected for correlates of protection studies. So all later phase studies should have such samples collected, like MTBVAC is doing that, etc. Even the H56 that didn't work has samples where one can identify correlates of risk now, of relapse or recurrent disease. So those should be done. And then there are multiple huge international cohorts that could be exploited. I think, for example, even the HIV cohorts, there are lots of such cohorts that we haven't really dug into carefully. Now, I know that it's always risky to go after the fact, to go to a data set or to a sample set, because you don't have all the data. But I think there are opportunities for us to look at that. These studies are expensive if you do it prospectively, but perhaps you can look at these other cohorts and see whether there are samples available.
But going back to the microbiome issue, because I've just been thinking, is the mechanism, could it be, so you mentioned some of the correlates when we were talking this today, and you mentioned natural killer cells and some of the interleukin and chemokines. So could microbiome changes kind of alter the immune system as such that you turn off and turn on stuff that could affect, just how would it work?
There are some really nice studies coming out now, just in mice. If you BCG vaccinate mice versus not, you look at changes in the microbiome, you can see differences in the proliferation and expansion of some species. And they've done this for HPV and for HCV also. And at least the current thinking is, so the microbiome speaks to the immune system using a variety of different mechanisms, and one of those is bacterial-derived peptidoglycan fragments. So the mechanism is still being worked out, and some of these fragments actually make their way to the brain, and they turn on neurons and they turn on pathways that stay on for a while. And so it's multifactorial. You change the composition of the microbiome that allows bacterial components to signal and interact with the immune system. And some of those effects last a very long time. And so you can think of really, one of the things we've been thinking of is developing reporter cell lines to actually look, for example, nod induction, and how they sense, how these nod receptors sense peptidoglycan, and kind of developing that you can use as another way of adding to the triage for different vaccines. But it's very early days. I think it's quite exciting to think around this. Yeah.
Actually, I want to ask you a political question. So I've been living this U.S. federal government cuts for the last 2 months. Sorry, I'm a letter. I work in the faculty office. And considering that figure that you showed about the large reliance on NIH funding for funding TB research, where do you see us going in the threat of the withdrawal of funding? And obviously, both HIV and TB research needs to continue. It's not something that we need to stop. But what are we going to do?
So PEPFAR and USAID funding is already stopped. This is more downstream stuff, okay? And already, I think Wits has already fired 1,700 to 1,800 people because of that. It's very clear from our discussions with Jabek and Tusi and people at NIH that all NIH funding is going to stop to South Africa. It hasn't happened, but it's going to happen. So we are going to have we're going to be hugely short of funds in South Africa to do the research that we think is important. I don't know what to say, except that this may also be an opportunity for us to diversify and to look at other sources and do things differently from before. At the same time, I have a faint suspicion that this, and American colleagues say this, that this is not a permanent situation, that a lot of this funding is ultimately going to come back. But in the interim, yeah, I think we're in a hugely precarious position. In South Africa, to compensate for the NIH, if you do not know, to compensate for the NIH losses, there are 2 applications going on to the Treasury, the one from Department of Higher Education, something like 2 billion rand to compensate for some of it. And they've got a proposal in. And then the MRC, with various other partners, is applying for 400 million rands, which then, if it is given, will probably be matched by the Gates Foundation, the Wellcome Trust, hopefully INSERM, and Novo Nordisk Foundation. So hopefully another 2 million rands, 2 billion, sorry, rands to compensate at least for some of the losses from this in the interim to really fund the critical infrastructure that will get lost, be that HR infrastructure or other infrastructure or junior investigators that are completely reliant on NIH funding.
So thanks very much, Willem. I'm sure for all the early stage investigators in the room, you can see that you have a future in TB research. We've heard that the pipeline is atrocious. We have a vaccine that might become 30% efficacious, and that we still don't understand what the correlates of risk or protection are, and why we see, this people get infected, they clear, or they go on for disease. So for all of you who are wondering what you should be doing, and I would put it to you that we still have a lot of discoveries to do in TB, and the story is not over. So if you are wondering what you should be doing, there you have it. We need vaccines, we need correlates, and we need better vaccines, a better understanding of the mechanism, this understanding the bimodal distribution, and understanding all the elements that drive TB infection on our continent. So there you have it, and we want to thank Willem for this, because you've really, in a wonderful way, kind of shown us, what we need to do, and what are the things that we should be thinking about at the university. So thank you very much.